Hello everyone, and welcome back for more human physiology. Last time we discussed a lot of commonalities between several different types of signaling methods, and arguably the most important commonality there is that every type of cell-to-cell signaling that we talked about will involve some type of signaling molecule binding to and activating a receptor on a target cell. So let's explore that relationship just a little bit further here. So all the different types of signaling molecules that we talked about, whether it's a hormone, a neurotransmitter, or a paracrine or autocrine signal, they fall under this kind of catch-all term that we call ligand. So a ligand is any type of molecule that binds to and activates a receptor. So the idea here is that there are a lot of different types of ligands. There's hundreds of different types of hormones, neurotransmitters, so the total number of ligands is going to be well into the thousands, right? But for any particular receptor, only a few or maybe even just one ligand will be able to bind to and activate that receptor, meaning most of the ligands out there will not be capable of activating that receptor. So the way I've always liked to think about this is it's kind of like a lock and a key, where a lock is going to represent the receptor here, and a key will represent a ligand. So think about the lock that opens the front door of your house or your apartment. So obviously there is a particular key that opens it, but there are millions of keys out there in the world, right? But there's only one particular type of key that is going to open your lock. There may be keys out there that look a little bit like your key, but there's something special about the shape and the grooves in your particular key that perfectly suits it so that it can open your particular lock. So if you look at this picture here, you'll notice that this receptor, this lock, has a certain shape to it that can only accommodate a key that has that matching shape. And under here with this key that says correct ligand, you'll notice that the shape of the key exactly matches the shape of the lock. Whereas the incorrect key here, the incorrect ligand, it has one part of it that matches, but another part does not. And ligands are going to be very similar to this. There may be some ligands that resemble other ligands, but generally speaking, it only takes a very small structural and shape difference to make all the difference in the world in terms of whether a ligand can activate a receptor or not. So a lot of ligands may look like certain other ligands, but only the correct ligand is really going to give you the full desired response from a particular receptor. So the other issue that we want to address in this hopefully very short video here is knowing what we know about the relationship between a ligand and a receptor. An important skill that we want to develop is we want to be able to predict where a receptor will be located on or in a target cell based on the chemical nature of the ligand. So this is why, dating back to chapter 2, we wanted to get a lot of practice in identifying what does a hydrophilic molecule look like and what does a hydrophobic molecule look like. What does a polar molecule look like? What does a nonpolar molecule look like? Because this is going to be how we answer this sort of question here. So if you will recall, dating back to chapter 3, when we discussed the cell membrane, its permeability, what sorts of things can cross it and what sorts of things cannot, it turns out that most ligands are not going to be able to cross the plasma membrane by simple diffusion. So what that means for those ligands is that the only logical thing is that their receptors have to be located on the surface of the plasma membrane, meaning that little binding pocket that accommodates the ligand will have to be facing out towards the extracellular fluid. So receptors like these are going to tend to be membrane proteins. So the conclusion we can make here is that if your ligand is hydrophilic or lipophobic, those two are the exact same thing, then that ligand's receptor will be located on the surface of the cell. So somehow, if you look at this picture here, your hormone or neurotransmitter or whatever else, when it binds to this receptor, something is going to happen that causes the internal physiology of the cell to change such that we eventually get our desired physiological response. Now, that is going to be a topic for the next video when we talk about signal transduction. So in more rare cases, usually when you're talking about steroid hormones, you can find the receptor inside the cell, either in the cytoplasm or inside the nucleus itself. So this is only going to be true if your ligand can cross the plasma membrane by simple diffusion, meaning that your ligand is going to be hydrophobic or lipophilic. Again, those two are the exact same term. So really the only case that this is going to be true is if you're dealing with a steroid hormone like testosterone or estradiol or progesterone, cortisol, aldosterone. So the only way this is going to be true is if you're dealing with a steroid hormone, or the only exception to that is thyroid hormone, which we will talk about in greater detail in Chapter 17. Speaking of Chapter 17, now that we kind of have a general feeling for what types of signaling molecules have their receptors where and how, in Chapter 17 we will get into better details on this when we start talking about specific hormones, and this will be an important skill for us to have in terms of identifying where those hormones' receptors are located. All right, so that is going to do it for this fairly short video. Go ahead and join us next time, and we will start talking about the idea of signal transduction. What that means is essentially what happens after the ligand binds to the receptor. How does the binding of the ligand to the receptor translate into a change in cell function that will eventually give us our physiological outcome that we want? So I will go ahead and sign off for now. See you later, and bye-bye.